Okay, so as I was saying, we're going to review for the lab practical, and then also we can talk about the exam material as well. But first of all, let me make sure that we know exactly what labs you're responsible for, because we kind of mixed them up a little bit with those newer labs. From the old lab manual, from the one that you downloaded, you're responsible for labs 1, 2, and 4. And in lab number 5 in the old lab manual, where we did the dialysis tubing, we didn't really do it as a lab. Remember, we just kind of put those bags into the beakers and let them soak. I do want you to look over that particular part of the lab. Let me pull up the lab manual so you know exactly what I'm talking about. So we're in lab 5. I probably have them sectioned off in such a way that I can tell you exactly what. Let's see, where is it? Yeah, it's part A1. And you can see down there, there's a table. Remember that we didn't fill in the table, but we put in the bags, and we found out, of course, that in most cases they gained water. And so you should kind of know what that was all about, how the water moved into the bag, or why the water moved into the bag, and what substance actually was able to move through the bag. Remember, we put, starch inside the bag, and now that wasn't able to move out because it was too big, but the water was able to move in, and that has to do with osmosis. So make sure you look over that part. Again, don't worry about that formula about the percent mass change of the bag. I'm not concerned about that. We could have waited before and waited after and then calculated that information, but I was really just interested in demonstrating to you the principle of osmosis. So just know kind of the basics of what we did in that particular part. So that wasn't really a full lab. And again, you don't have to worry about anything else in that lab that we didn't do. So, so it's 1, 2, and 4. We did not do 3, so don't worry about 3 in the old lab manual, and then that little bit of 5 that I told you about. Now, for the other ones, the newer ones, the LabQuest ones, we did LabQuest number 2 and 3, and I must have those in here. Hold on a second. 1, 2, 3. There's 2. I'll come back to this page. So 2 is the one we did the other day with the bullion cubes. Okay. I'm not going to make you, run the Data Logger Pro software. There's nothing in the lab practical that's going to question you about that per se. But you should, again, know the overall results of the lab and what we found out. And remember, this lab is a good lab for illustrating those 2 points that we talked about in class. This is the fact of diffusion. The things freely diffuse. They go from a higher concentration to a lower, which is why the bullion cubes were dissolving. Remember, the bullion cubes represented solute, whatever they put in, the salt, chicken parts, whatever they put in there. Those are all solutes. And when you first drop the bullion cube in there, they're all concentrated in that one area. And that's a disequilibrium, which Mother Nature does not like. So by simple diffusion, that passive process that we discussed, the solutes are going to begin to disperse. They're going to begin to diffuse out into the beaker. All right. The common way, it dissolves, right? It dissolves in the water. That's what dissolving is. That's diffusion. That's diffusing. And remember, the other concept of that tied into was what we talked about when we did the very first lab, when you had to measure the blocks of wood and calculate the surface area to volume ratio. And at the end of the lab, we were able to determine, looking at this table here, and I don't recall the exact numbers. You may have to calculate some surface area and volumes in the lab, but I'll talk a little bit more of that when we get to the review of the lab practical in general. But remember, what we were doing in this particular lab is we were trying to determine the relationship between those 2 variables, right? We were trying to determine the relationship between the surface area to volume ratio, which is what we call the independent axis. It's the independent axis. It's called that because that's what you as the investigator were manipulating. You were changing that. You were dropping in 1 piece, or a piece that was in a half, or 4 pieces in quarters, or whatever, as you see on the far left-hand side over here. And then the rate, still haven't figured out what micro S's are. It's probably some measurement of solute concentration, something to do with conductivity. Let me look that up right now while I'm thinking about it. Conductivity, what does micro S mean? That was what the meter was doing. That's how the meter was telling how much stuff was in the water. It was actually measuring the conductivity through the water. Right. We'll call it. That's a micron. Huh. I'll have to try to find out what exactly that means. But nonetheless, it is a rate of diffusion, right? And that was what we had on the y-axis. And remember, we found out that in the surface area to volume ratio, as the surface area to volume ratio increased, I think we had something like 0.4, 0.6, I think, wasn't it? Maybe that was 0.5, 0.7, and 0.8. I believe that's what we had. So as the surface area to volume ratio increased, we found out that the rate of diffusion increased. Remember, it was what we call a positive relationship. In the fact that positive relationships are those that as one variable goes up, the surface area to volume ratio goes up, the rate of diffusion goes up. And relaying that back to the cells again, remember, that's why cells stay small. Cells stay small so that things can very easily diffuse across their membranes, things that they need, waste products they need to get rid of, and all those things we've talked about. And the smaller the cell is, kind of in this case, the smaller the bullion cube was. When we get down to 8 pieces of bullion cube, each one was really, really tiny. The smaller the pieces are, the bullion cube, therefore the smaller the cell is, the faster rate of diffusion that's going to occur, okay? And of course, since we know that cells need diffusion to remain alive, to move things across the membrane, then makes sense that evolution and selection is actually going to select or going to fashion smaller cells. The smaller the cell, the higher surface area to volume ratio. The higher the surface area to volume ratio, the more efficient the cell. Okay, so that's kind of what I want you to know about that lab. Again, I'm not real concerned with you knowing the the aspects of using the data logger software. So don't worry about that too much. Let me go into, while I'm doing this, let's go into lab 3 and look at that. Lab 3 was the one, again, not the lab 3 in your lab manual, the old lab manual, but the new one that I had. Remember, this was the first one we did with the data logger. This was the one we did with the acids and the bases. And let me see, there's a couple things in here I wanted to point out. I want to make sure you guys, must be on the next page. Let's see. Here we go. There we go. This is what I want you to know. I want you to be, first of all, you need to know how to calculate change in pH. And that's simple enough, right? So the pH at 30 drops minus the pH at 0 drops. And then the buffering ability. I do want you to know the formula for the buffering ability. The buffering ability formula is right there. Okay. So remember the little delta. So we're not just subtracting the pH, we're subtracting the change in pH. So we could do an example for it, all right? Let's say that we were testing a particular substance and we were dropping in drops of acid, and after 30 drops, the substance went from a pH of, let's say 4, and it dropped down to a pH of 1, okay? So it went from an acidic solution to a much more acidic solution. Remember the way the pH scale works. The further you move away from 7, the closer you get to the ends of the scale, the stronger the acid or the base is. So something has a pH of 1 is a much stronger acid than that which has a pH of 4. So again, the first thing you have to do is calculate the change in pH, and that's simple enough. 4 minus 1 is 3. And then if we dropped in a base, okay, so this would have been the acid change. And then if we took the same material and we were doing drops of base to see what the pH was after 30 drops, again, it's the change in pH. So again, we might start at 4, right? That makes sense because it would have been the same solution. And when we were done, the pH was now, 9. Okay, so that's a, over here, change of 3, change of 5. All right, now to figure out the buffering capacity, or to figure out the buffering ability, the formula is very easy, right? You take the change in pH of the acid and you subtract that from the change in the pH of the base. So the change in the pH of the base was 5. The change in the pH in the acid was 3. So the buffering ability is 2. Now it doesn't have any units. It's really only a number that makes sense when you're relating it to other numbers. So if you went up to someone and you said, oh, this has a buffering capacity of 2 or buffering ability of 2, it doesn't mean anything unless you're talking about how does that relate to other things that you tested. It's kind of like saying and going to someone and they say, oh, how tall or what's your height? And you go, tall. You know, yes, but you're not giving enough information there, obviously. So, you know, you do need to know how to calculate buffering ability. I may also have a list of different things that have buffering abilities and actually ask you to determine which one has the greater buffering ability, and that's just going to be the greater number. After you subtract those numbers, this number here, in this case 2, the higher the number, the higher the buffering capacity. All right? I'm not going to ask you what materials were better buffers than other materials. Don't worry about that. I'm really kind of more interested in you understanding what the idea of the buffering capacity was all about. Let's do here. That all selected like that. Anyway, let's see. Let's look at the questions. How should the pH? Where is the one question I was interested in? Oh, here we go, number 5. And I'm sorry, folks, I don't know why it's got this selected looking thing on here. Let me see if that helps. No. All right, look at number 5. The second part of the question. Why was it beneficial to include the plot of the water in acid and the water in base with every experiment? In other words, what was the purpose of doing it to the water and then testing the different substances? What was the water acting like in our experiment? You may remember we talked about that. Yeah, it's a neutral, it's a control. There you go, right? So we were going to compare the differences in the buffering abilities or the changes in pH of those different substances, where we're using the water as a baseline. Because remember, water is not an acid nor a base, right? Water has for our purposes, water has a neutral pH. It's right in the middle. So we were using that as our baseline, okay? And we were comparing that to other things. So water, like you know, where we talked about in experiments before, was something you were using as a control, and a control is something you can compare something to. All right, so you might be able to compare the buffering ability of that material to the buffering ability of water, right? So you always have to have a control, and that's what the water was doing. Water is not an acid nor a base. In this case, it's obviously just simply a control. All right, so that's those 2 labs. Anybody have any questions on that? On those 2 labs? Go back to where I had this thing here. I know you can't see the writing on this. This was something I had to import from an older version of Collaborate, but this is just kind of giving you an overview of kind of what the lab practical exam is like. Like I said, I know a lot of you may have not ever taken a lab practical exam, and so there's a bit of anxiety associated with just what exactly is a lab practical exam. When you come into the room, and let me remind you, I won't be there next week. You will be proctored. The lab technician will be there, you know, proctoring the lab exam for you. But when you come into the lab stations will all be set up around where you normally sit. Now, you won't be able to come into the lab early, right? Come in there and sit and wait for lab to start. Everybody will be outside. The lab tech will let you all in at the same time. Pick any station that you want to sit at. All right, doesn't matter. You can sit at the one you usually sit at. You can sit at any of them. It doesn't matter to me. You're going to eventually go through each one of them. So the way it basically is set up is they're going to be, you can't see, there's 24 stations. Each station has 2 questions. A couple stations have 3 questions. The idea is there's 50 total questions, okay? And each question is worth 3 points. Now, in some cases, the questions are written in such a way that you might be able to get partial credit. In some cases, it's not. For example, if I give you something like a true-false question, you know, either it's true or it's false. You can't get partial credit on that. But on those in which you can get partial credit, if you don't know the full answer, just make your best attempt at trying to figure out what it is, and you might get a point or 2. All right, so 50 questions, 3 points each, 150 questions. And I can't see what the last one does, but I believe that's the amount of time. If I remember right, that's the amount of time, and it's going to be 1.5 minutes per station. So the lab technician will give you 1.5 minutes to do your to answer your question. And again, don't let that bother you in thinking about enough time. You will have enough time. The way the questions are worded, they're worded in such a way that it shouldn't take you more than 1.5 minutes to answer those, even if it is that. There may be some very simple calculations, but a lot of the questions are just simply, do you know what this is, or do you know how this works? All right, so that's kind of a simple one or two word answer, maybe a short sentence at the most, but there's no questions in there that are going to ask you to write a 75-word essay or something like that. So a minute and a half will be plenty of time to do each of the stations. Now, if the lab tech calls time, in other words, at the end of a minute and a half, what you'll do is you'll stand up and move to the next station. Everybody will. That's what I mean by you're all going to go through every station. If he calls time and you're not finished yet, right, here's what I suggest you do. You're going to have a piece of paper. It's old school. You have a piece of paper. Now, the only thing you need to come to class with that day is something to write with and a calculator, not a phone calculator, an actual calculator. If you don't have one of those old calculator things, don't worry. He's got 4 of them in there that you can use. So there are calculators available, but you don't need anything else. I'm not going to let you use your notes on this. I know I talked about this the other day, but I decided there really isn't that much material. And to be honest, you really shouldn't need your notes for what I'm going to be asking on the lab practical. You should have all of this pretty well enforced in your mind. So I don't think you'll need those. But if he does call time, if he says, all right, time's up, move to the next station, don't panic if you haven't finished yet. Write down the question or write down something that sparks you to what the question is asking. Like, for example, if the question says, name a material, name an organic molecule that contains an amine and a carboxylic acid group. Okay, remember we talked about there was certain molecules out there that were very easy to identify. Their name of the actual monomer, the molecule, was reflective of the 2 functional groups that are found there. Remember we talked about that's amino acids, right? The building blocks of proteins. Amino acids are called amino acids because they have amine functional group. That's the one with nitrogen. Make sure you know those functional groups. Don't forget those either. And a carboxylic acid. So it's amino acid. Now I could reverse that question, right? And I could say, name a molecule that has a, like I said, amine group and a carboxylic acid. And for some reason you draw a blank, you just can't remember what it is. Jot that down. Make yourself a little note. Say amine plus carboxyl, and write that on the side right next to the question. Right? You move into another station, and maybe very quickly you answer that one. So you got maybe 45 seconds or a minute. You can go back and look at that question again. Now, you can't go back to a station. All right, you can't go back to a station. Once you've gone through them once, you're done. But that doesn't mean that you couldn't bring some of that information with you if you're not finished yet. If you're asked to do a calculation and your time runs out, write down all the information about the calculation. Write down all the variables, and then in another station, perhaps you can come up with that answer. All right? Those are just a few little techniques. Keep in mind, too, that a lot of times, like in exams, even in lab practicals, you can use that concept of test mining. Remember I was telling you about that? That sometimes an answer or a hint or something that might spark your memory about a particular concept might be found somewhere else in the exam. And that could be the same thing with the lab practical too, okay? All right. What I'd like to do now is I'm going to go through and just kind of some sample questions just to give you an idea of what you might expect to see. I guarantee you're going to see a microscope. You may see, you obviously more than one microscope, but you're going to see a microscope, and I'm going to tag parts and ask you to identify them. Now, this is the picture straight out of your lab manual, and all those little gray arrows are, in identifying the parts. So you should know all the parts of the microscope, and by all means, please don't neglect to remember what the parts do. All right. So, for example, and what it'll be little dots, like things you might see people put on garage sales when they put the price, like little colored dots or something like that. And I might say, okay, the orange dot, orange color. Oh, I do. The orange dot, I've tagged it right here. All right. And then I may just say dot A. It doesn't matter. And then down here, I've tagged this piece. And then let's say I'll take a blue one and I'll tag this right here. Okay, and you'll have to know what those are. All right, so let's do that. Let's do a little practice here. The orange dot, what is the part of the microscope that the orange dot? It's not multiple choice, Sadie. There is some fill in the blank, but most of it's short answer, 1 or 2 words kind of thing. Oculars. There you go. Yeah, Marie, oculars. Okay, very good. Oculars or ocular, either one. Oculars is plural. I only tagged one, but I wouldn't nick off points for that, obviously. How about the yellow dot? Now, I may say in these questions, be specific. So look at where the yellow dot is. It needs to be a little bit higher than where I drew it, by the way, folks. But look at where that is and what part of the microscope am I pointing to there? There you go. Right, Shaniqua. Perfect. Not just focus. Not just focus, right? Fine focus. And I could easily put that dot on the knob behind it, and you'd tell me it was the coarse focus, right? Okay. Now, the blue dot. It's an objective. Okay, tell me if I said specifically what objective is it, what would you call it? Remember, we had 3 objectives. Each one had a name. Let's go through them then. Here's the smallest one right there. It's got the red ring around it. That's a 4x objective. That's the lowest power objective we have. What do we call that lowest power objective on any microscope? Think about what you do with it. Yeah, you guys got it. That's the low power objective. Right. Thank you. I'm sorry that came in a little bit later. I was already talking. All right, but back to the one with the red ring. What do we call the lowest power objective? A scanning objective, right, Colin? Think about what you do with it. You scan. Remember when you were using your microscopes the other day, you can't just simply put something under the microscope and slap the highest power objective and expect to see something, right? It's too magnified. You want to stand back for a second to see your image. So you're going to scan across the slide with the lowest power objective. Now, in our microscopes, that's 4x. That's not the case in every microscope, but every microscope has a scanning objective. Every microscope has that first objective. You may not see a lot of detail, but you're looking for something on the slide. You used it when you were looking at the different protozoans, right? When you scanned around till you found something, and then you went to the low power objective. And then after that, which objective did you go to? What's the one with the blue ring? What's that called? Okay, perfect. Now, let's do a little bit of mathematics. The low power objective, the one with the blue dot, has a magnification of 10x. If I asked you on an exam, if you sat down at a station and had a microscope set up, and it was the low power objective that was clicked, that was pointing straight down. In other words, it was as if you're getting ready to use the low power objective. I might ask you, as the microscope is set up right now, with the low power objective in use, and the oculars that you have on that microscope, what is the total magnification? There you go. That's right. Now, how did you do that? What's the 2 things that you needed in order to do that? Remember, magnification is it. There you go. There you go. Yep. You always take the magnification of the objective lens and multiply it by the ocular. Now, this is an easy question, folks. You really only don't have to worry about the oculars. All of our oculars and all of our scopes are 10x. Don't worry about looking at the oculars, turn around, wasting your time on that kind of stuff. Because, again, it's a timed exam. So think about doing those things that are reasonable. I'm not going to switch oculars on you or anything like that. It's going to be a 10x ocular. So very simple. Whatever objective is clicked into place, just take the magnification of that objective, multiply by 10, boom, you're done, right? You're ready to move on. See what I mean by sort of short answers? And then maybe a function. Can't really see it, but underneath the stage, there was a device. You caused it to go up and down. I don't know if that's the knob right there. There was a knob that you turned, and that object moved up and down. Now, first of all, what was the name of that object? What was the name of that object that moved up? Remember, it was like a little lens of light that was coming through from below. What was the name of that? And I'm going to give you a little way to remember it too. When you're looking through the stage and you see that little lens and the light is coming through there, notice what that thing is doing. It's taking the light. Nah, Marina, they're under there too. I'm sorry, but those are the two vertical knobs over here on the right. I'm talking about that piece, this piece that I've circled. It can move up and down. And like I said, if you're looking through this part, you could see that piece move up and down underneath the stage, and it had a lens, and the light from below, this light source, was coming through there. What was that little lens doing to the light that was coming up from below? Remember, I told you, using microscopes, it's all about the light. You got to have proper lighting, and you got to have ways to adjust that lighting to see the image. Well, it didn't so much make it darker or brighter. It was more a matter of contrast. Remember, the little device that I'm talking about, the function of it was contrast. Contrast is a little different than brightness and darkness, right? You played with your monitors or anything like that. You know what I'm talking about. Contrast kind of gives you, increases or decreases the difference between the dark and the light. Okay, so now we know the function. It adjusts the contrast. But what was the name of that device that does that? If I told you that it focused the light from below, that's why you could see that little lens what I'm pointing to with the arrow. If it's focusing the light onto the slide. So what's another word for focusing? It is a light adjustment, Marie. That's right. But what's another word for focusing? What's another word for concentrating? Because that's what that thing is doing. It's acting like a hand lens. When you were a kid, you know, you could burn a piece of paper with a hand lens because you focused the light. So what was that device called that did that in this case? It's focusing the light from below onto the slide, so you get a maximum amount of light on that slide. There you go. It's the condenser. Again, relate what the name of the part is with what it does. The condenser focuses or concentrates the light, right? That's why you saw that little bit of light coming through that small lens pointed to with the arrow. That's what it was doing. It's taking the light from below, which is more diffuse. It's, you know, and it focuses it into a peak, and it allows concentrated light to act upon your image. You see, as again in microscopes, it's all about the light. You got to have good, proper light in order to see the specimen. And if you move that condenser up and down, what it did is it changed the amount of light that was focused, and that's what adjusts the contrast. Okay? All right, so go through that page in your lab manual and do kind of what I've done here. Take a piece of paper or something and blanket over the answers, and just go through and identify each of the parts and identify what each of the parts does as well, right? How about this? How about the arrow right here, this arrow? Well, let me erase all that other stuff. What's this arrow pointing to? What's that part? There you go. Good. All right. Easy enough, right? Any questions on that? And it will be your same microscopes. It won't be other microscopes from other places. It'll be exactly the same. Good deal. Okay, now this is not something we did in lab, but it's something that we had to be aware of in lab during the microscope lab. Recall I told you that as we go from the scanning power to the low power to the high power, the diameter of the field of view. Now, y'all remember what I mean by the diameter of the field of view, the DFV. If I asked you to define that, if I said, what is a microscope's diameter of field of view? What is that? Again, think about what the word is saying. It's the field of view. Yeah, it's what you see. Yeah, it has a size. That's right, Sadie. It has a size. The diameter is going to be the size of what you see. Yeah, the field of view is when you put your face up to the microscope and it looks like I have a mask on. When you put your face up to the microscope and you're looking into the scope, that circle of light that you see, right? That's the field of view. Now, what does it mean that it gets smaller as we go from scanning to low to high power? Does that mean that that circle that you're looking at gets smaller and smaller in the microscope? What does that mean that it's getting smaller as we go up in power? Now, there you go, R. Thank you. Zooms in. That's what it does, right? When you go from the scanning to the high power, the reason why we say the diameter of the field of view gets smaller is what R just says. It zooms in. So that's, once again, why you would not use the high power objective to look at a microscope slide to begin with. Imagine that this is a microscope slide and you were going to use the high power objective. Right away, you'd made yourself, I don't know, drop a pond water or something like that. So you're going to scan around and see what you can find, but you don't realize that you're supposed to use the scanning objective or using the high power. Because it's so zoomed in, you basically are going to have to search little tiny areas back and forth across that, sample that you got to see something. And not only that, at high power, your depth of focus is very thin. Now, what was depth of focus? These little drawings down below are intended to sort of give you a visual. This is the field of view right here. So that's you looking down into that circle of light. So what is the depth of focus? Notice what the little cylinders are doing. Yeah. The depth of focus, it's getting narrower, but what is the depth of focus? Yeah, I mean, what do we mean by that? Remember I talked in lab about using the high power objective? Yeah, kinda. Remember I said in lab, though, using the high power objective, not only are you trying to search across that slide with zoomed in, which is really hard to do, but that when you're trying to focus something, you can, with the high power objective, you can easily go in and out of focus so quickly you don't even notice the image is there, or the specimen is there. All right, let's take the slide now and look at it from the other angle. Let's look at it from the side. So here's the slide, and here's the cover slip. I drew the cover slip a little bit higher. We're really zoomed in here just to kind of give you an example. Let's imagine that here's our paramecium, right? It didn't look like it. It looked like those paramecium were living in a two-dimensional world, right? But they weren't. They're living in a three-dimensional world. The third dimension is very short, but nonetheless, it's still there. So when we're using the scanning power objective, as we're scanning around and looking down on this slide, if we're using the scanning, we have a very wide view. We also have a lot in focus at the same time. So there's a better chance we're going to see that paramecium. Now, if we were using the high power objective, what would happen? I'll draw this over here now again. There's the slide, or the cover slip. Here's the paramecium. If you're using the high power objective, notice how much shorter the cylinder is there. That means if you're using the high power objective, and remember at that point you're only using the coarse focus knob. You never use the fine focus knob. You never use the coarse focus knob when you're using the high power objective. Remember, the high power objective comes very close to the slide, so you don't want to use the coarse focus knob because that moves the stage a lot. We don't want to break the slide. But you notice that cylinder of light that you're looking at when you're in high power is very thin. So when you're searching around for that paramecium with the high power objective, it's just like if you were looking at it from above, this one up here before. You're looking at little tiny pieces as you're trying to search for it. When you're trying to do it with the high power, the depth of focus means you're searching three-dimensionally a very narrow sliver. So you might actually pass right over top of that paramecium, but since your depth of focus is so narrow, you might be focused above the paramecium or below the paramecium. So in other words, like Ara says, it won't be in focus. Yeah, Amber, that's actually a pretty good way of saying it. It's how deep you're looking into it. So as you go up in power into the high power, you're looking less deep. You're looking more shallower. So, and then finally the light, right? As you go up in power, a function of the physics of microscopes, of lenses, the way they work, is the fact that the amount of light that passes through the lenses diminishes as you go up in power. So high power objective, the image is going to be darker, okay? And a low power objective, the image is going to be a lot brighter. Sorry, I had to stand away. So I'm saying the high power objective, it's going to be darker. Now the image turns darker. You don't turn it darker, but it becomes darker because of the way the lenses are set up. You have to turn around and have to add more light to it, okay? So if on the exam I was to ask you, as we go up in power, as you increase the magnification, what are you going to have to do light-wise to make sure that you still see the microscope? Well, there's another really short answer question, right? So I'm saying, okay, you're viewing an image, you're going up in power, you're going scanning low to high. You're going to have to focus it and all that sort of stuff, of course. But all that aside, what are you going to have to do to the light to make sure that you still see the image? You have to make it brighter. You have to turn the light up. Okay? All right, very good. Okay, so those little cylinders down below, if you can kind of relate the way they look to those three parameters and how they change, it might help, right? So you notice the cylinder is getting smaller in diameter, it's getting narrower in depth of focus, and it's getting darker. Okay? All right. Some formulas you need to know. People say, do we need to memorize some formula? Yeah, there's a couple. It's not too many. There's one right. Get rid of that one. Don't worry about that. You did need to know C1 V1 equals C2 V2. Oops, hold on 1 second. I'll be right back. Sorry about that. I forgot that I left the back door open when I let the dogs out, and I needed to go close it. Not exactly interested in air conditioning the state of Florida. All right, so know those formulas, C1 V1 equals C2 V2. So what I thought we would do is do a couple of examples of that. And you will see some of these on the exam. This is where you would need a calculator. And remember, again, like I told you, that you will have calculators there. Let me get the screen a little bit easier for me to write on. Okay, first of all, let's define the variables again. What is C1? What's C1? Anybody? What's C1? Got to know what these variables mean. The concentration of what? It is a concentration, or you're correct, but the concentration of what? Right, concentration of the stock solution. All right, so we'll say stock. Okay. What's C2? It's obviously concentration. Yeah, that's the ending concentration. That's the concentration you want to make. Remember, the way the problems are worded is you start with a stock solution of X amount of percent concentration of dye, and you want to make a solution that has a less concentration of dye. So this is your ending concentration. Your stock, your ending concentration What is V1? Maria? Barbara? Right, V1 are, yeah, that's what you need to go. Now, when you're making a dilution, the V1 is you're going to go over to the stock solution, you're going to take your little pipette, and based on your calculations, that's how much you're going to draw out to get exactly the concentration at the end you want. All right, so that's your variable. That's what you're asking for. You've got the stock, right? You know what its concentration is. You've got C2. You've been told, well, you don't have C2, but you've been told what C2 is going to be. What's V2? Somebody else as well. Come on, all of you. Yes, that's how much you want to end up with. You got it right. So V2 is the ending volume. That's why it's called V. It's ending volume. So a question like this could be worded, okay, you start off with a stock solution of 80%. You want to make 15 mLs of a 30%. All right. Now, like any word problem, whether math or biology, whatever class you're in, at least for me, the best way to ensure that I would do a word problem correctly is I always wrote down the formula and I always read through the problem more than once just to make sure I understood it. And then I went back and picked out the pieces I need. So what did I say? Let's do that one. Let's say we're going to start with an 80% concentration. Okay. We want to turn that into a 30% concentration, and we want to make a total of 15 milliliters of that. Okay, so I tell you what, why don't you guys, whoops, doggone it. Sorry, folks, wrong button. You got 80% concentration, you want to turn it into a 30, and you want to make 15 mLs. All right, so you got your formula. Let's plug in what we know. All right, what's C1? What's the concentration that we're starting with? Okay, very good. What's V1? No. That's V2. What's V1? Nope, that's C2. V1 is X. There you go. V1 is X. That's what you're solving for, right? And C2? What's the concentration you want to end up with? There you go. And how much of that do you want? There you go. See how easy that is? So the answer to that question is X equals 30 times 15 divided by 80. Let me get my handy dandy little calculator here and figure out what that is. Wake up, computer. All right, so 30 times 15 divided by 80. All right, what'd you guys get? There you go. Very good. So now, what is 5.625, Sadie? What is that telling you? What do you do with that number? That number indicates what? Remember, you were solving for V1. Okay, so what the ending volume was 15, Kirian. The ending volume is 15. 5.625, that's how much you're going to go to the stock solution, this 80% stock solution that's sitting on a table somewhere in the lab, and you're going to take your pipette and you're going to withdraw 5.625 milliliters. Now, we wouldn't be able to do that with our pipettes. They're not that fine, but there are pipettes that do go down to the 10/100,000th of an ml. So you would draw 5.625 mLs. You would put it into your beaker, your flask, whatever the case may be, right? So that's the dye you need. But 5.625 mLs of stock, but we're wanting to make a total of 15. And remember, the whole purpose behind this is diluting it. So if you went and you get 5.625 mLs of stock and you put it in a beaker and you say you're done, you've done nothing. Done is withdraw 5.6 mLs of stock. the concentration of that little tiny bit of stock that's in your beaker now is still the same. You're wanting to dilute it. So what do you add to it to dilute it? What are we going to dilute that stock with? What did we dilute them with in class? Yeah, water. And how much water? Is that the difference between 15 and 5.625, Colin? I'm going to assume it is. Yeah. Okay. Yeah. So, Colin, tell us how you did that again. How'd you come up with that number? Right. See, you're not done with the 5.625. What I said is, how would you go about preparing the dilution? When you would draw on 5.625 mLs of stock and put it into a beaker, you've diluted nothing. You've got to come behind and add 9.375 mLs of water. 9.375 and 5.625 is 15. Remember, it's got to add up to V2. So be very careful with those problems when you're doing an exam. You don't stop with just solving V1, because the problem is going to ask you, how do you prepare the dilution? Okay, I take 5.625 mLs of dye. I put it in of the stock, excuse me. I put it into a beaker. I add 9.375 mLs of water. That adds up 15 mLs, and that's a 30% solution, right? So there's kind of two parts to that, to doing those problems. One is solving V1, okay? And then the other one is adding enough water so that V1 plus the water equals V2, right? So your answer V1, and it's usually water that we use for dilution, plus, X amount of water, whatever it turns out to be, that has to equal V2. Remember, that's what we want to end up with. We didn't want to end up with 5.625 mLs. We wanted 15 mLs. Okay? Now work through those problems in the back of lab number 2, right? And this is the one in your actual lab manual that you downloaded. The answers you remember at the back of the book, and hopefully you've been doing that already. If you're having problems with those, you need to let me know, okay? So we can work on that. It really has a lot to do with simply just kind of practice, to be honest with you. You do a half dozen or so of these, and you'll be fine and ready to go. All right, any questions on that? Anybody need me to do another one of those? Everybody okay with working out those kinds of problems? Okay, now, in your study problems in the lab manual, I did throw you a couple of curveballs. Like in a few of them, I gave you, say, for example, the measurement of the liquid was in liters, all right? And I wanted you to express an answer in milliliters. Or maybe I gave you your answer in milliliters, and I wanted you to express the answer in liters. Let me tell you the best way to prevent making a mistake. If you read the problem and I'm starting off with one unit, let's say liters, and I want to end up with another unit, let's say again milliliters, do the conversion first. If you're going to end up as milliliters, take that liters and convert it to milliliters first. Now you will have to do some metric conversions on the exam too, right? So if you have a way that you've been doing for years, like I said in lab, by all means use that. If not, use that little mnemonic device that I told you guys about, right? Kids have dropped over dead converting many blankety blank metric blankety blank numbers, right? You can write that right across the top of your answer sheet when you come into the lab practical. So do the conversions first. You have less chance of making a mistake. So if you know you're going to end up with your answer in milliliters, but you're starting with, centiliters or liters, do the conversion first, then work the problem, okay? Speaking of that, what if you came up with an answer, let's say 35.3 milliliters, right? Just for some particular situation. How many cubic centimeters is that? How many cubic centimeters is 35.3 milliliters? This is right out of lab 1. Those of you who might be doing any calculations right now, don't. You don't need to calculate anything here. You remember, this is why we could tell the volume of the rock. Well, hold on, Colin. Not 3.53. Nah, you're converting. Don't do conversions. This is how we were able to tell the volume of the rock. Remember, we dropped the rock into the graduated cylinder. Maybe we started off with 60 mLs. The rock, when we dropped it in, now we have 75 mLs, and the difference is 15 mLs. You don't talk about a volume of a rock in liters, right? Liters is for liquid. You talk about volumes of solid objects in cubic centimeters or cubic millimeters or whatever the case may be. But remember, there's an interesting relationship in the metric system, one that doesn't exist in the English system. In the English system, you have to do all these kinds of conversions, but it's really nice in the metric system. 1 ml, I'm going to type this right in here. 1 ml equals 1 cubic centimeter. 1 ml equals 1 cubic centimeter. All right, same question again. 35.3 mLs is how many cubic centimeters? There you go. No calculations, no conversions, nothing at all. Now, if I went the other way around, same thing. If I said 35.3 cubic centimeters is how many mLs? 35.3 milliliters. Okay? This has been doing lab practicals a long time. One of the most common answers that people miss, and I'm like, why? It's a one-to-one relationship. There's no conversion. I think people just get confused. So remember, in the metric system, a milliliter is equivalent to a cubic centimeter. If you think about it, a measurement of volume, like liter, right, is a three-dimensional measurement, right? And where a centimeter can be one-dimensional. Centimeter, centimeter squared, centimeter cubed. All right, we live in a three-dimensional world, so a milliliter, a volume, is equal to a cubic, whatever linear measurement we're talking about. 1 ml, 1 cubic centimeter, okay? Speaking of that, and while we're on it, there's also another interesting relationship that occurs in the metric system between, let me see where this is, so I can give you a reference point. It's back in lab number 1 again, and it was right at the top of page 10. No, not the top, but below table 1.9. There's a little piece there in italics, and I said the density of water is 1 gram per ml or 1 gram per cubic centimeter. Because remember, mLs and centimeters are the same thing, right? mLs and centimeters are exactly the same thing, so you don't have to do any conversions there. So what if I told you, okay, I have 14.75 mLs of pure water. How many grams does that weigh? Why 13, R? Kelsey, what about you? No, I said 14.75. I'm sorry. Or did I say? I might have said, I'm sorry. It's 14.75. Shayla? Amber, what do we got here, guys? 14.75 mLs weighs how many grams? 14.75 mLs of water. I have to say water weighs how many grams? Yep, that's right. There's another easy one. See what I'm talking about? Some of the questions on the lab practicals, it'd be something like that. How long can that, as long as you remember that, and you're not trying to do any conversions or do anything mathematical, as long as you remember those two basic ideas, an ml is a cubic centimeter, pure water weighs 1 gram per cubic centimeter or ml, because it's the same thing. Boom, your answer's done, you're ready to move on, right? All right, any questions on that? Of course, once again, don't worry about this one here. How about this formula here? All right, that was the formula that was in the 4th lab, the one we do with the microscope lab. It's the same type of formula as the C1V1 formula, but what we're doing in this case is we're relating the magnification of an ocular with how big its field of view is. Remember, we use the diameter of the field of view. We use how big that circle is to help us estimate sizes. I'll talk about that in just a second. But we know that there's a direct ratio between the magnification and the diameter of the field of view. So let's set up a problem. M1 is going to be your starting magnification. M2 is going to be, we'll call your ending magnification. I don't know if that's the perfect word, but you'll see what I mean in a minute. DFV1 is the diameter of the field of view for this magnification. Okay. And DFV2 is going to be the diameter of the field of view for that magnification. Okay, so this is basically the same way as before. I'm going to give you 3 variables. You've got to solve for the 4th. So I might say, all right, let's see. We've got a microscope. The magnification of an objective is 20x. Now, we don't need to worry about putting the x. We'll just put the 20. So magnification 1 is 20. I know that the diameter of the field of view, how do I know that? The manufacturers told me, or I've taken, they have little rulers, actually. You can measure them. But I know that the diameter of the field of view at 20x is, let me think, 5 millimeters. Okay. All right. Now, on this microscope, I also have an objective that's 50x. And I would like to know, because the manufacturer hasn't supplied it to me, or I don't have a way to measure it, I would like to know what that diameter of the field of view is, because I have some objects that I want to measure under the 50x. All right, so diameter of the field of view, that's what you're solving for. I give you the magnification. I gave you, I have to give you 2 of these. I have to give you 2 parts of the equation. I either have to give you M2 DFV2, or I have to give you M1 DFV1. And then again, all it is a matter of substituting, rearranging the equation. And in this particular case, what we got is 50x equals 20 times 5. Right. So x is 2. We always want to make sure we put units. Y'all agree with me? Yeah. All right, now do a little reality check when you're doing these problems, because remember, what you do know about microscopes is as you go up in power, the diameter of the field of view should get smaller. So is that what happened here? Did the diameter of the field of view get smaller as we went from 20 to 50? Good. Check yourself on that. You know, check yourself on that and say, well, wait a minute, we went up in magnification, but the answer I got was actually bigger than the original one. I've done something wrong, right? But it's one of those things I always call do a reality check, right? Stand back for a second and say, does that make sense? I know all along from all on my microscope work that as I go up in power, what's going to happen is the diameter of the field of view is going to get smaller, and yet I got an answer that was bigger. I must have done something wrong. Okay, so make sure you know those 2 formulas. There is one other formula I would like you to know. It's very simple. It's density is mass over volume. If I ask you to calculate the density of something, well, in anything that I ask you to do measurements on, I need units or you're going to lose points. All right, so for example, the density, typically the units are gram per cubic centimeter or milliliter. But pay attention to the way the problem is set up. Make sure you put the units. If you give me a density of 5, or just give me a number of 5, I don't know what you're talking. 5 oranges, 5 apples, 5 miles. I need units. So make sure you put units. Okay. All right. Any questions on any of that? All right. Don't worry about that. That was from a previous lab. Don't worry about that. Here we go. I'm going to put models of functional groups out there. Number 1, is this a functional group? And if it is, tell me why it is, and if it isn't, tell me why it isn't. Is this a functional group? Yes or no, and why? It is a functional group. Okay, R, which one? Right, carboxylic acid. And is that a functional group? And if it is, right, how do we know it's a functional group? How do we know it's not just a molecule? There you go. Remember, a functional group is something you add to, one of those big carbon skeletons that we talked about in the chapter about organic molecules. So on an exam or on the lab practical, you're going to see some functional groups that you have to identify. And you might actually have a bunch of molecules built and you have to pick out the functional group. Very simple, folks. Don't try to identify each molecule. Look for the one with the open-ended bond. Now, there is one functional group that will have two ends that are open. Which functional group is that? It's going to have two open bonds on it. Perfect. Yeah, because remember, the functional group for the ketone is like that. Now, if I ask you to identify a functional group and I say be specific, you might either see this one or this one. Those are both carbonyls. But if I ask you to be specific, you need to tell me specifically which one it is. The other one that we just talked about was the ketone. What's this one? What's this one that has that terminal hydrogen? There you go. Aldehyde. Nice, nice, nice. Good deal. Now, all the functional groups. You might have to identify, this is just straight out of your lab, you might have to identify functional groups. I might say, all right, and I might have an arrow pointing and say, what is this functional group right there? Or I might say, look at that molecule and name one of its functional groups that it has. So what is that one that I'm pointing to? What is the name of that? It's the simplest functional group. Yep, that's a hydroxyl. Very good. All right, up here. We didn't do anything exactly like this, but we did do something, where we talked about acids and bases and that sort of stuff. We can determine the pH of a solution using like the probe, like we did in that one lab. We can also determine the pH of a solution by using what we call a qualitative indicator, right? I think I've talked about this in class before. A qualitative indicator is something that gives you a quality. Like, for example, you could ask the question, is this solution an acid? A qualitative indicator would say yes. It wouldn't tell you what the pH is. That would be a quantitative indicator. A quantitative indicator, like those probes, give you a quantity. Yes, it's an acid. Its pH is 3. Okay, so qualitative and quantitative indicators are very different. And one of the qualitative indicators that we use in science is color change. So, for example, in the material that you see there in the beaker, in the one on the left-hand side, if it's yellow, it's an acid, and if it's red, it's a base, or it's neutral. And in the purple one that you see there, if it's purple, it's a base, and if it's green, then it's neutral. Those are qualitative indicators, right? Those are indicators in which you can tell yes or no, it's an acid or a base, but you don't get a number. An example of a qualitative indicator outside of the lab would be John is taller than Fred. That's a qualitative indicator. John and Fred might both be very short people, but all I'm saying is compared to one another. So a qualitative indicator needs a comparison, right? John is taller than Fred. Now, if I turned around and I said John is 6'2", and Fred is 5'8", then it's a quantitative indicator, right? Know the difference between quantitative and qualitative indicators. Easy way to do this. Easy way to do this. Don't worry about reading through whether something is qualitative or quantitative. If numbers are being reported, it's quantitative. Simple enough, right? Okay, here's a block of wood up against a ruler down here. How long is that block of wood in centimeters? In centimeters, how long is it? Okay, how long is it in millimeters? Millimeter. There you go, 31. Right? You got to do your conversions. All right, so know how to use a ruler, measure a block of wood. And I might use that as some sort of a staging area to ask you about doing some conversions. Okay. Any questions on this page? All right, here you go. There's a graduated cylinder up here in the upper left. What is the volume of liquid? Looks like it's probably water in that graduated cylinder. Make sure you read the scale very carefully. Remember, not all the lines are marked, so it takes us to figure out what the ones that aren't marked, what they mean. I will tell you for this particular graduate. Oh, there you go, Jessica. Thank you. Anybody else? You all agree with Jessica and Amber? Do you see where they got 21.5? Does anybody not see where they got 21.5? There's 20, 25. Each one of these must be 1. There's 22, 23, 24, and each one in between must be a half. Okay. What's that curved part called? You notice how the water's curved there? What's that called? Yep. And which part of that do you read? Do you read the part that's elevated on the ends or the part in the middle? Yeah, you read the lowest part of the meniscus. There you go. All right, very good. Okay, so you know how to read a graduated cylinder. Good. Now let's use that graduated cylinder to calculate the volume of an irregularly shaped object. It's easy enough to get the volume, unless they're bullion cubes, right? But it's easy enough to get the volume measuring a block, length times width times thickness, and you're all ready to go. But what if it's something like this fish? So take a look at this fish and tell me what its volume is. Now, let's see if I can zoom in a little bit so you can see the numbers. There we go. All right, so this is before and this is after. Before we put the fish in and after, I'd like to know what is the volume of the fish. Okay. This is 30. Each line in between is 1. There's 40. Does that help? There you go. Not 38. 38 is what the water is raised to when you put the fish in there. Remember, this is like putting a rock in there. Same thing. The water was originally 32. You put the fish in, and now it's 38. So what is the difference between those two? It's 6. What's that difference made up? That's got to be the fish. Remember, when you immerse something in water, it displaces a volume or displaces an amount of water equal to its volume. Think about what that equation is saying. An object that's submerged, I think this is in first lab as well, an object that is submerged, a fish, a rock, will displace the amount of liquid equal to its volume. Equal to its volume. So we start off with 32. You put the fish in, it's 38. The difference is 6. The fish is the 6. Okay? Now, what's the unit we're going to use? The fish has a volume of 6 what? Yeah, you got to subtract them. There you go. What's the unit? 6 what? Yeah, but not technically, right, Amber? You wouldn't report the volume of a solid object like a fish using liquid, right? Liters is liquid. Grams is weight. Looking for volume. That's all right. So amber's on the right path, but we wouldn't use milliliters. We'd use cubic centimeters. There you go. Perfect, right? Because remember, up above, I write that. Why does this work? Well, it works because we know that 1 ml of water is 1 ml is 1 cubic centimeter. So however many milliliters were displaced, and in other words, displaced, the water rose, that's how many cubic centimeters a solid object is. I don't know if you've ever noticed when they talk about ships, ships are oftentimes the size of ships are reported as a displacement. What that means by that is when I put the ship into the water, what is the weight? They do it with water, not volume. They do it with weight, not volume. What is the weight of water that's been displaced? It's been pushed aside by putting that ship in there. So if a ship has an 11,000 ton displacement, that means when you take that ship and you put it in the water, it displaces enough water that weighs 11,000 tons, right? That's what that means by that. And it's the same thing working here. We would say this fish has a 6 square centimeter displacement, all right? Or 6 square centimeters of volume, and that's how much water was displaced, 6 mLs, okay? I already talked about density being mass over volume. What are these little things called on the outside of the paramecium? No. That's what that's called. Cilia. Cilia are shorter. Organisms tend to have many cilia. Flagella are longer. Organisms tend to have fewer. In this case, you see the Euglena there has got 1. Actually, Euglena have 2. You can't see the other one, but they've got 1. Okay. Now this organism down below, this amoeba, it moves around too. Right, cilia and flagella for movement. I'm sure you know that. But what are the name of the structures that the amoeba uses to move around? They don't have cilia or flagella. What do they use? Pseudopodia, there you go. Fake feet. Yep, that's what these things are. Pseudopods. Pseudopodia. Excellent. Nice job. Okay, any questions on that page? All right, well, I'm going to the next one. Think about this for a second. I put a material into a beaker, and what that material does is it causes the pH of whatever's in that beaker to stay the same. Okay, so no matter what I add to that beaker, that pH is exactly the same. What was the name of that material that I added that caused the pH not to change? What do we call those things that do that? Yep, buffers. Don't worry about that. We didn't do any of that. No, don't worry about that. I have that on the exam. So this is what type of relationship? And this one? What's the other one called? Going this way. Negative, or sometimes a negative too, by the way, is called an inverse relationship. And sometimes a positive is called a direct. All right, good enough. Don't worry about what this is. We didn't use that. Oh, here we go. This is a bunch of circles. What I would have you do is draw me out an atom. All right, let me get an example of one. How about let's see, what do I need? I need let's do aluminum. Let's do aluminum. Aluminum is an element. Aluminum has the atomic number 13. Okay. So aluminum has how many protons? Okay, how many electrons? How many electrons? How many neutrons? What's the matter? How many neutrons does this aluminum right here have? know that for sure? It's unknown, right? It's not given. What else do you need in order to figure out neutrons? What other number do you need? You need the well, you already have the atomic number. That's that right there. Mass, right? That's a number that's often written in the upper left. And for aluminum, the atomic number, or the, excuse me, atomic mass is that. Now, how many neutrons does it have? How many neutrons does it have? Marie, look at that again. There you go. Subtract the atomic number from the atomic mass, and you're off and rolling, right? Simple enough. Okay, now let's take these circles up here, and let's diagram out where the electrons are found in aluminum. So I might give you a sample element and have you answer questions about valence electrons, valence shells, or I might have you draw one out. So let's do that. How many electrons can go in that very first shell? The smallest circle. The black dot is the nucleus. Okay, 2. Why is that, Jessica? Why can you only put 2 electrons in that first shell? What is it about that first shell that only allows 2? No. Remember orbitals? How many orbitals in that first shell? No. How many orbitals in that first shell? 1 orbital, and each orbital can hold 2 electrons, okay? How many orbitals in the next shell, the next circle out? Right, each holds 2, that's correct. So how many electrons in that, how many orbitals in that next shell, the next larger circle? It's not 2. It's not 3. It's 4. Yes, you have 1 spherical and the 3 dumbbell-shaped orbitals, right? Sorry, I can't. There we go. So you can hold a maximum of 8 electrons in that next shell. So let's put 8. What do we got? 10 so far. The atomic number of aluminum is 13. 13 electrons. Where's the other 3 going to go? Where's the other 3? Where should I put the other 3 electrons? In that last shell. There you go. And what is that last shell called? That outer shell is called what? It has a name for it, a special name. It's a valence shell. So how many valence electrons does aluminum have? How many valence electrons does it have? Got 3, the ones in the outer shell, okay? All right, take a look at that. I know we're getting ready to run up on time here, folks, so but take a look at this picture on the right. You sit down at a microscope, and the low power objective is the one you're looking through, not the high power, the low power. Not looking through the scanning, the low power. And you see a paramecium, and it's situated off to the side of the circle there. And I could ask you a question. When you go to the high power objective, what do you have to do first to make sure you're going to see that paramecium when you go to the high power objective? What are you going to need to do? You're going to center it. Absolutely. Yeah, it's perfect, right? You got to put it in the center. Remember, the diameter of the field of view gets smaller. All right, if I ask you to calculate the size of this paramecium, and I told you that, well, let's see, this is 0.4 millimeters is the diameter of the field of view. All right, or 400 microns. Yeah, 400 microns. So it's 400 microns all the way across. How big is this paramecium? Just give me a rough estimate, folks. See how well I can. You'll see what I'm doing. Right? You line them up, head to tail. Is that 0.4 divided by, well, it's 0.4 divided by 4, or no, wait. No. Jessica, think about that again. If the diameter of the field of view is only 0.4 millimeters and the paramecium fits it inside the field of view, It can't be bigger than the diameter, right? Or how did you calculate that? Yeah. No, 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 no. Why by, not by 400, right? You took the diameter of the field of view, and what did you divide that by? I know it's 10:50, folks. It's actually a little over. If you need to leave, by all means, do that. I've only got just a couple of more minutes. If you want to stick around, I will record this. You can come back and listen to it later. But all right. So you took the diameter of the field of view, and what did you divide that by to get the size of one of those paramecium? You see what I'm doing here? I'm saying, all right, I'm estimating how many could I fit all the way across the diameter. It looks like I could fit about three and a half. Yeah, Amber, by all means, please. Yeah, 3.5. I would agree with that. So you take the diameter of the field of view in millimeters. Usually in microscopes we deal with microns, right? So let's just go ahead and do the conversion. It's 400 microns divided by, what'd you say, 3.5? There you go, 3.5. Because it looks like 1, 2, 3.5 of them will fit across, okay? Now, if I wanted to do it widthwise, then of course you would just do it the other way. Okay. Now, I will tell you how big the field of view is. You don't need to worry about that. All you need to know is the process by which to do it. That's okay. You got any questions about it now, or you understand how to do it? Cool. Here is, let's say, pH. No, wait. Yeah. No, temperature. Enzyme reaction rate. Enzyme rate. Goes up, drops. 2 questions. First of all, where would you find the optimum? Now, you can draw on the whiteboard if you want. So if you have the ability to draw on the screen, point to or make a little line where the optimum would be. Or you can tell me. You know, obviously presume there would be numbered. There you go. Whoever drew that, thank you very much. That's the optimum. So then you would look down here on the x-axis, and that's what you would answer. If that was 80 degrees, then that would be the optimum. All right, now tell me why the reaction rate increased as it got warmer. That's right. Speeds up the reaction. Okay, very good. But then as the temperature got higher, why did the reaction drop so quickly? What happened there? Denatured. You denatured the enzyme. Excellent. Excellent. All right, that's my last slide, but I have one thing I want to talk to you about. Let's take 3 objects, X, Y, and Z. And I'm not going to write the units only in a matter of time, but it's going to be units of density. So grams per ml or grams per cubic centimeter. Okay, so that's going to be the units for all of these. The first one is 3.8 grams per cubic centimeter. The other one is 0.3. And then we have one that's 1.9. Okay. Which one of these is going to float in water? Will any of them float in water? Or will they all sink? Or will they all float? Remember in lab 1, you got to go back and look at that relationship about water and how much it weighs. 1 gram of water, all right, is 1 ml, right? 0.3, right? That says that if the density of water is 1 gram per ml or cubic centimeter, that's the density of water. So I'm putting whatever Y is into water, and it weighs 0.3. So it's less dense, it'll float, and then X and Z, both of those will sink, won't they? Right? Any questions on any of that, folks? Anything you need me to go over again? Talk about some more? It's up to you. Again, if you do need to leave, I understand. I will be recording the rest of this, but if anybody has any other questions, we can certainly do those too. For the exam. Did I ask an essay question? Let me look. Sometimes I ask essay. Oh, wrong button. Oh, God. And sometimes I don't. So let's see. Oh, yes, I did. There you go. Okay, now you don't know what the cellular transport mechanisms below are because I didn't give you the whole rest of the question here. But what I'm talking about here is being able to tell the differences and the similarities between diffusion, osmosis, active transport, passive process, active process, facilitated diffusion, exocytosis, receptor-mediated endocytosis. This is all at the end of the membrane chapter when we're talking about how things move across the, well, it was most of the membrane chapter. So again, I love compare and contrast questions. And in a compare and contrast question, the best way to study for it, make yourself a table. And even on the exam, if you want, you could list it that way. And that table says, how are they the same and how are they different? So if I'm asking you, let's say, diffusion and active transport, how are they the same and how are they different? Okay, well, they both move materials across the membrane. All right, simple enough. That's how they're the same. Now, where they're different in quite a few ways. Diffusion goes from higher to lower, and it's passive, doesn't need energy. Active goes from lower to higher, it's active, and it needs energy. And active transport also requires a carrier protein. Okay, diffusion does not. So that would answer that particular part. So what I'm going to do is I'm going to throw together 3 or 4 cellular transport mechanisms, like I just gave you the example, and you're going to have to tell me the difference between them in terms of the way the materials move, whether they need a carrier protein, whether they need energy, whether it's an active or a passive process, which is essentially the same thing. Okay, that's your essay. Yeah, you can use them on the lecture exam, Jessica. No notes on the lab practicals, but the lecture exam you can. All right, folks, if you don't have any more questions, then I'm done for the day. I'm in office hours, obviously, so if you have any others, but other than that, you're welcome very much. You all have a good weekend. I will see you next week online on Wednesday. Remember, of course, your exams on Monday. You have your lecture and your lab exam. Okay? You do your lecture exam upstairs in room 216. Thank you, Laura. Hi, Amber. Are you still there?